Okay, hi, good morning, everyone. So let's continue talking today about how our cells handle moving things across the different membranes that make up the cell. So mainly we've been focused on discussing the outer cell membrane of the cell. And remember, we learned that all the membranes inside the cell, whether it's the outer cell membrane or the various different internal membrane structures, are all made of a phospholipid bilayer, right? Two layers of phospholipids embedded with all kinds of other interesting molecules, things like proteins, for example, that do all kinds of functions, as well as other things like other lipids like cholesterol that serve to maintain the integrity of the membrane. And also cholesterol can be used as a building block for making other types of important signaling molecules like hormones that we will talk about in a subsequent lecture. Last time, we spent considerable discussion on diffusion, right? And understanding how if you have a membrane and you have solutions on either side, for example, water, right, containing all kinds of solutes in it, different dissolved molecules, the concentration of a particular solute on one side, right, can form a gradient. If the concentration is very high on one side of the membrane and lower on the other side, then the natural tendency or direction that the solute will want to travel or flow is down this gradient from the high concentration side to the low concentration side, right? From high to low. And that's intuitive. I don't think we need to really discuss any real deep scientific basis for that because we just sense that in everyday life. Things want to go from high to low concentration. And if you have a membrane getting in the way, right, that might just in some cases, just slow down the ability of those molecules to move through. If those molecules are very small and maybe nonpolar, they can just diffuse simply right through the membrane from high to low. Or we discussed last time, sometimes the solutes in question are maybe larger and cannot just flow through the membrane themselves. They actually have to have their movement facilitated or helped out. So we looked at last time examples of different proteins that might be embedded in the membrane that act as things like channels, right, or carriers to allow things to be able to move from one side to the other, still in a downhill flow from high to low, right? But importantly, that type of facilitated diffusion, right, requires the involvement of things like proteins to create openings or opportunities for those particular molecules to flow through. And hopefully you realize that the shape of those proteins can be very specific to match the shape of the particular solutes that are being moved. All right, so last time we did say that there are situations when our cells have to move things against the gradient, right? In other words, moving things on purpose from one side that's low concentration to the other side of the membrane where there might already be a very high concentration of that particular solute, right? So going against the natural flow, not surprisingly, would require some energy to actually force this to happen, right? And so that's what we're going to talk about today. Important examples of active transport that our cells engage in. All right, so let's start off with just getting back on the same page about the importance of water. Right? All of molecular cell biology, whether it's explicitly mentioned or just implied, right, takes for granted the fact that all of these molecules we talk about are reacting in a water environment, an aqueous environment that makes up both the inside of our cells and the outside of our cells. Right? Already numerous times this semester, I've tried to stress or emphasize the importance of thinking about how all of these molecules that we look at are surrounded by water. Right? And the very fact that these are the molecules that we study, because these are the molecules that are important for life on Earth, has to do with the fact that evolution took place, right, in the context of liquid water. So it's a good opportunity to go back and think about this again. If you consider the effects that having dissolved solutes in water has on the pressure of the water, that's something to really importantly keep in mind. So what do I mean? If you dissolve something like table salt, here's a teaspoon of table salt, sodium chloride, right, that you drop into a glass of water, stir it up, and the salt crystals easily dissolve, right? We no longer see them as crystals. They just become dispersed in the water. Consider that any solute like this sodium chloride, right, takes up space in the water that was in the glass. Before, that space or that volume was entirely just water, but now that you've mixed a whole bunch of salt in there, these salt atoms, the sodium and the chlorides, they take up some of that space. Right? I mean, if you added a ton of salt in the water, the level of the water would rise, even though the salt is invisible because it's dissolved. Right? So these solutes, any kind of solute dissolved in water, would take up space that otherwise the water molecules themselves would be occupying. Right? So the relative water concentration, right, per constant volume would be dropping as you're taking up more and more of that space with something like the sodium chloride. And so sodium chloride, this is an interesting example here. I think from general chemistry that sodium chloride is an example of ionic bonding, not covalent bonding. Remember, covalent bonding is when two atoms share electrons in orbitals, right, in order to keep those electrons happier at a lower energy state. And as a result, that's a very strong bond that forms between two atoms. Right? Ionic bonding is different. Remember, you probably learned in general chemistry that ionic bonding has to do with one atom giving up an electron and another atom happily taking that electron. Right? So in the case of sodium chloride, that's what happens, right? The sodium atom gives up an electron, and in the process, it becomes more positively charged, right? And the chlorine atom happily takes that electron, right? And in the process becomes negatively charged. And when that transfer happens, you end up with a positively charged ion, a negatively charged ion, right? And since they were just right there next to each other when this exchange happened, they tend to stay together because now the positive and the negative charged ions, right, want to stay together. But again, they're not sharing the electron anymore, right? So therefore, that attraction between the plus and the minus of these ions is much weaker than a true covalent bond. So in a crystal of sodium chloride, the ions are arranged this way, right? And there's opportunity for water molecules to get in between them. So here you have the crystal of salt in the water now in the glass. And what you see is, again, it's surrounded by all these little water molecules, and the water molecules are bumping into the sodium and the chloride ions, sort of knocking them off, sort of getting in between, right, the positive and negatively charged ions. So you can see this sodium ion is happily then surrounded by sort of like a shell of water, where the water's oxygens, because remember, water is polar, so the more negatively charged oxygen side of the water molecules are attracted to the sodium ion. And over here, right, on the right, the opposite. The chloride ions attract the more positively charged hydrogen on the water, right? So again, forming these shells of solubility around them. And that's why the table salt dissolves so easily, right? And then these ions would be interspersed all throughout the liquid, through the water. So here are a couple of terms that I want you to be familiar with. Osmolarity, then, is concerned with the total solute concentration. In other words, how much salt is in this saltwater solution. So the osmolarity of the solution would increase with more salt being added. The other concept is osmotic pressure or osmotic potential. That has to do with the fact that these water molecules then are being displaced because they have to share the same volume of space here with now all of these dissolved solute molecules. So there will be a tendency of pure water, pure H2O, to cross a semi-permeable membrane, right, to flow into a salt solution like this in order to equalize the total water concentration on one side of the membrane versus the other. So remember that in our cells, there's all kinds of solutes, all kinds of things are dissolved in the water that make up our cells, right? Not only ions like sodium, like chloride, like potassium, but also big molecules like the proteins, right? And so you better believe that big globular proteins take up a lot of space that otherwise could just be space taken up by pure water if those water molecules were in there uninterrupted, right? So this is another opportunity to get us thinking about the shape of proteins. Remember that, so here are different ways of showing this globular protein here. This is a soluble protein, meaning in this particular example, this is a protein that happily can dissolve in water and just float around as a single globular protein, right, surrounded by water molecules. Remember, this is still, in this picture here, a single polypeptide chain of amino acids, right? Let's say, for argument's sake, that this end over here, right? So it shows this illustration shows the peptide backbone as a ribbon, right? So let's pretend that this end of it over here, I should point this way to be more consistent with how the slide is shown, right? This might be the amino terminal end of the peptide chain, and then this would be a whole chain of amino acids that have some secondary structures to it, and it goes around and around and around, sort of like a maze if you're playing it kind of like a game. And eventually, I don't know, maybe this is the carboxyl end sticking out over here, right? So one long string of amino acid residues that have folded up into a complicated tertiary structure. And then this figure is also nice because it shows you the space filling diagram if you were now to take into consideration, right, all the atomic orbitals of things like the R group side chains that are hanging off of all the amino acids as you go along this polypeptide backbone. So that's a space filling model. Look at this figure in the middle, this green and red one. What they've done here is they've actually highlighted, right, where are the amino acids that tend to be hydrophobic amino acids, in other words, having hydrophobic side chains, versus the amino acids that are hydrophilic, have hydrophilic R groups, like the polar or charged side groups, right? So you can see, as the protein folds up, there will be a tendency for the hydrophobic residues to be sort of pushed into the middle, and the hydrophilic residues to be happy, sticking on the outside. Again, what's driving this, right? Water is driving this, because the water molecules that are surrounding this protein, right, will push the hydrophobic residues to point inwards, while the water molecules will happily hang out and play with the hydrophilic residues on the outside. So again, remember, this resulting three-dimensional shape has to do with the particular sequence of amino acids, so that those amino acids, when they're chained together and interact with water, will fold up exactly in this shape, right? And what determines that chain of amino acids? The original gene in your genome that specifies in the DNA sequence, right, what the RNA sequence should be that's copied from that gene, and then that RNA, after it's edited, right, the RNA sequence, every 3 RNA bases will code for a particular amino acid to be linked together by your ribosome to make this, right? And then because this is all happening in water, the water molecules interact with it and cause it to form its right shape. So this third illustration with the animation, I like this, right? Because this actually lets you now imagine the fact that, yes, right, all this is happening in the context of this protein being in water. So all the little water molecules are surrounding it, right? I often say the illustrations that we look at don't bother having the water molecules indicated, because it would just be too confusing, right? You wouldn't be able to see anything going on. But it's fun to think about, yeah, because it's surrounded by all these water molecules, the water molecules are interacting with the amino acid residues. So if this protein started to unfold and maybe some of the hydrophobic residues in the middle, like the red ones in the middle diagram, started coming out, what would happen? The water molecules would just keep bumping into them and shove them back in, right? So without water, this protein would not be the right shape. Without water, this protein would not work to do whatever your cell needs it to do. Okay. So let's talk about water now and water moving across the membrane, right? So here we have in purple an illustration of a semi-permeable membrane. Maybe this is something like your cell membrane. We can call it your cell membrane for the sake of this discussion. Let's say on the right side of the membrane is inside of the cell, right? And inside the cell, you'll have a lot of things like proteins. So this particular tan-colored molecule, right? Let's say it is a protein, just like the globular protein on the previous slide, right? Because it's dissolved in water, we can call it a solute, right? Because it's a solute that's floating around in water. And then the little blue balls are the water molecules. Right on the other side of this membrane, maybe on the outside of the cell, you would have fewer concentration of things like proteins, right? So what does that mean? On the right side of this membrane, right, the water pressure, or the amount of pressure that's actually due to the number of water molecules, is lower than on the outside. Why? Because that amount of space that the water has, right, is being taken up by all of the other dissolved solutes, like these proteins. So per volume, you'd have fewer water molecules in here compared to on the other side. So if this is a semi-permeable membrane, meaning that maybe the water molecules can flow through very easily, as shown here, right? But proteins cannot, what would happen? You'd create a situation where you are concentrating the proteins on the inside of the cell, right? And remember, we said life is all about being able to concentrate certain macromolecules, right, to really encourage, right, the efficiency of different biochemical reactions to happen that maybe wouldn't happen if things were just allowed to kind of spread out and disperse on their own, right? Your cell has to concentrate certain macromolecules together in order to drive, right, or favor certain reactions happening. But that means that the water in this case would flow from the right side or the left side of the figure into the right. Now, this might seem kind of counterintuitive, right? Because you might say, well, on the right side, right, it's more crowded, right? So I wouldn't imagine that the water from the left side would really want to kind of like add to that crowded space, right? But the thing you have to keep in mind is that, yes, the water molecules will want to flow from left to right in order just to even out the number of blue water molecules on either side of this. What does that mean? That means the volume on the right has to increase in order to accommodate the water molecules flowing in. Right? So already you can imagine then that a living cell has to deal with this, right? And again, living cells cannot just rely upon things flowing back and forth purely by concentration gradients. They need to somehow have a level of control in order to survive. So here's some extreme cases of what happens if that control is lost. Right? The fluid in our bodies, right, is of a particular salinity, or in other words, a particular salt content, right, that is the appropriate physiological content for our cells to be able to deal with things like how to manage water going in and out of them. And so this quality of a solution or an environment in which you're talking about a cell being placed, right, has to do with the tonicity of the environment surrounding the cell. So in normal situations where you have, let's say, red blood cells in normal blood, right, that environment, that solution surrounding the cells is an isotonic solution, right? So this would be what you'd find in your normal tissues. That means the interior of the cells and the exterior of the cells, the fluid on the outside, right, have very similar solute concentrations in terms of things like salts. As a result, your cells, like these red blood cells, can maintain their normal correct shape. However, if you drop those red blood cells, right, into a hypertonic solution, meaning now, like the salt water that you've dropped them into has way more salt than normal, right, what would happen? The water that's inside the cell would want to flow out of the cell to try to even out the amount of water molecules, right, on either side. That would cause your red blood cells to collapse, right? They would shrivel up, as seen in this photo here. The opposite would be, what if you took these poor red blood cells and dropped them into something like pure water, right, that doesn't even have any salts in it, right? As a result, this would be a hypotonic solution, hypo meaning low, right? So the tonicity of the solution is way lower than inside the cell. In this case, the water molecules from the outside of the cell would rush into the cell to balance out the relative solute concentrations, and these poor red blood cells would swell up uncontrollably and would burst, right? So if you think about that, as you're sitting there, all of the cells in your body are bathed in this proper isotonic solution of body fluids, right? And that has to be carefully maintained in order for all the cells of your body to be able to normally function and carry out their different jobs. All right, so let's examine the tonicity of the different sort of compartments of fluid in our body. Now, you can imagine our body, of course, is made up of many different compartments, right? Tissues, organs, different spaces, things like that. But if you were to kind of functionally think about it as how many sort of distinctly different compartments of water do we actually have in our body from a functional standpoint, you might be kind of amused to realize that there's actually only really 3 functional compartments. In other words, anything that's inside any cell of our body, right, that's one compartment. In other words, all the water that's inside of our cells, contained by cell membranes, that would be what we call the intracellular fluid compartment, right? Inside cells, intracellular fluid, right? Everything immediately surrounding the cell, for example, in the tissue of wherever this cell is, right, that would be the interstitial fluid. So immediately surrounding one cell, between one cell and another cell in something like a tissue, right? That would be another compartment, right, inside of, let's say, an organ, for example. And then you have the third compartment, which is all the water that's flowing through your circulatory system, right? That's not really in any of your solid tissues. It's constantly flowing around, delivering all kinds of good stuff to your tissues, right? So for example, the plasma of your blood would be the third main compartment of water in our body, right? So if you think about it, yeah, it's weird to think about it this way, but there are 3 main compartments of water. So look at this, right? Not surprisingly, the relative balance of positive charged solutes and negatively charged solutes in a particular compartment are more or less equal, right? So that's what these bars are here. For each compartment here, right, you have the component cations, in other words, the positively charged ions that would be in this fluid, and you have the negatively charged anions that make up the negative sort of charged particle component of these fluids, right? So for each compartment, that's relatively balanced out, the positive charges and the negative charges. But importantly, right, I want you to notice this, the actual makeup of what positive charged ions, right, are involved and in what proportions in each of these compartments can be quite dramatically different. The cation and anion composition of our blood plasma is very similar to just the fluid that's surrounding our cells, right? Maybe that's not super surprising. So you can say for the most part, right, the interstitial fluid and your blood has a very similar makeup. Right? However, those are dramatically different than the makeup of the fluid inside of your cells. In particular, I want you to notice things like I mentioned before, inside the cells, you have a lot more proteins concentrated inside the cell. And I think you guys get that, right? Because what have we been talking about? That the expression of genes is all about making proteins, right? And the majority of those proteins function by staying inside the cell to carry out their functions intracellularly, right? So you'd have a higher concentration of proteins inside the cell, right? Compared to, let's say, in the interstitial fluid, right? Immediately outside of the cell, or even the blood plasma. You have more proteins floating through your blood plasma than you do necessarily in the space surrounding your cells, because many proteins are delivered through going through the blood to arrive at tissues and cells, right? But aside from proteins, look at these positively charged ions, right? Things like the potassium and sodium, right? Notice how inside of your cells, right, you have a lot more potassium in purple here compared to sodium, contributing to the positive charges inside of your cell, compared to right on the other side of the membrane, on the outside, it's flipped, right? You have way more sodium ions than potassium ions in the interstitial fluid, okay? So this right away should make you think, hey, this doesn't just happen naturally just by the flow of concentration gradients. Clearly, the cell, right, at the cell membrane is doing something to actively and intentionally create a disequilibrium of potassium and sodium, right, across the membrane. And this alone cannot be done by just simply letting these molecules diffuse, because if you just let them diffuse, it would just even out, right? So how does our cell manage, right, to maintain this on purpose disequilibrium? Well, here we go. So that's today's discussion, having to do with active transport. So again, right, on the left is what we just looked at on the previous slide. The fluid inside of the cell maintains very high potassium concentrations, but relatively low sodium concentrations. But immediately on the outside of the cell, the interstitial fluid, right, you have high sodium, right, and low potassium. So on the right here is an illustration of one of these proteins that handles this. So this is a transport protein, okay, that functions as a pump. Right? So rather than just passively allowing things to flow back and forth like a facilitated diffusion carrier protein would, right? Here, this protein complex actually has to pump things, right, in one direction. Therefore, it requires energy to do so. So this protein complex is made of several different protein subunits together, right? These purple alpha ones, there's also an orange beta one, and this blue gamma one. You don't actually have to know the structure of this, right? I want you to get the concept. What it's going to do is it's going to bind 3 sodium ions on the inside of the cell, right? And pump them out to the outside. And at the same time, it's going to take 2 potassium ions from the outside and then pump them into the cell. Right? In both cases, the sodium and the potassium are going to be moved or pumped against the gradient, right? Because you already have a lot of sodium on the outside. That's your big sodium contribution to interstitial fluid here. And yet the cell is still pumping more sodium out. And then the opposite is also true, right? That its cell is pumping potassium in, even though the inside concentration of potassium is already high. So this illustration is a nice one because it really helps you imagine how this is happening. So here we have a cell where you have the cell membrane going around, and we're examining the function of this, what we call sodium-potassium antiporter. It's an antiporter because it's transporting 2 different ions in different directions. So in the initial conformation of the protein, right, it's strongly stable with this conformation where it's open to the inside of the cell. Right? In that conformation, 3 sodium ions that are floating around in here, right, fit into the protein. Now, the protein also binds a molecule of ATP. When the sodium ions bind in there, right, the shape of the protein begins to change, and it actually breaks the 3rd phosphate group off the ATP, and the 3rd phosphate group now gets stuck onto the protein itself. So what have we discussed? What happens when you modify proteins by adding bulky groups to them, right? The polypeptide chain of the protein has to change its shape in order to accommodate the fact, right, that now it's a slightly different shape because it has this phosphate group attached to it. So the amino acids all kind of have to jostle around in order to get comfortable again. And what that does is it changes the shape of the protein to force those 3 sodium ions out. Now, an important principle to keep in mind here is those 3 sodium ions would not on their own want to go out, right? For example, if you go back up to step 2 here, imagine that there's a ton of sodium ions already out here, right? You can already easily, intuitively imagine then that if you have a ton of these gray sodium ions out here, the shape of this protein that's holding on to those 3 from the inside of the cell is not going to want to change its shape and face outwards. If anything, the sodium ions on the outside would keep this whole protein kind of shoved in this conformation, right? With the sodium ions still kept on the inside of the cell. So that means the amount of energy or the force of the protein changing its shape because it has this phosphate group stuck to it is a stronger force than the force of all the sodiums on the outside wanting to resist this happening, right? So that's the principle. So the phosphate addition is a stronger force that forces the protein to change its shape against what all the sodiums on the outside would want it to do. And as a result, it forces those 3 sodiums out. Right? At the same time, now when this antiporter is facing or open to the outside, that is the right shape for 2 potassiums that are on the outside to bind. Right? And likewise, already inside of the cell, there's a ton of potassium. So now you can imagine that this antiporter won't on its own want to change its shape to kind of force more potassiums into the cell. But this shape right here, right, at step 4, is only because there's that phosphate group still stuck to the protein. If you then release the phosphate group, then the protein will want to go back to its original shape. And that force, again, is stronger than any resistance that the potassium ions have inside the cell pushing up against it, right? So as a result, those 2 extra potassiums that came from the outside get forced into the cell, and then this resets. So the important thing to keep in mind here is the need for energy, right, in the form of ATP, breaking high energy bonds, making lower energy bonds, and the consequences of that on changing the shape of this protein. All right. So active transport is all about movement against the gradient. So this is really important because let's imagine this living cell down here, right? If you think about things like what gets moved uphill, right, across the membrane, things like nutrients. So obviously inside of a cell, you'd already have a lot of nutrients that are being used by the cell to stay alive, right? So the concentration would already be higher inside of the cell than the abundance of nutrients on the outside of the cell. And yet the cell still wants to take up as much nutrients as it can. So that's an example of active transport, right? To get more nutrients into the cell. Ions, like we just looked at, right? Being able to move potassium across in exchange for sodium, right? And also getting rid of waste products. A cell cannot have a lot of waste products in it, right? Because by definition, many of these waste products might be toxic. So the cell has to move waste products across the membrane, but it's dumping it right into the extracellular space here, the interstitial space that already might have a lot of waste products, right, from the other cells in the tissue. But yet the cell still has to move these wastes out. Okay, so all of these require energy. So let's discuss this concept of direct versus indirect active transport. So here in this cartoon on the left is a active transport protein, just like the ones that we looked at, right? They're called direct active transport because these proteins themselves manage to bind and break ATP, right? Changing the shape of the protein, allowing the protein to do something like in this case, in this cartoon, pump protons, H pluses, from one side of the membrane where the H plus concentration is low to the other side of the membrane where the concentration is already very high, right? And if it keeps doing this, right, these direct transport proteins maintain or build up this artificial concentration gradient of high protons on one side of the cell membrane compared to low protons on the inside. All right, so that's direct active transport, and hopefully that doesn't seem very confusing. The part that gets a little tricky is you have other proteins in the membrane, right, that can take advantage of that artificially created proton gradient, right? Because the protons on the outside of the cell would love to flow back downhill, right, into the cell in order to kind of balance out the proton concentration. So that's a very strong potential force that you can tap into. And so this protein over here, it taps into it. It is the right shape to allow these protons to flow back into the cell, right? And as it does so, it uses that force to at the same time bind and move some other solute, S, against that solute's concentration, right? So in other words, you are building up a high concentration of solute inside. Now, this could be, for example, nutrients like we discussed, right? And it does so, right, because this transport protein is just taking advantage of a gradient that was already set up with the protons. And again, you have to conceptually realize then that the force of these protons flowing through here is stronger than any resistance that the solutes on this side have in terms of wanting to resist more solute being moved in, right? And that's how it works. So we call this indirect active transport. Why? Because this protein is not binding and breaking ATP on its own, right? So you might say, well, then that's not active transport. It doesn't require energy. Well, but yes, it did require energy. It required the energy that was used by the direct transporter on the left to set up the artificial gradient, right? So you might say, well, that's complicated. Why would our cells do that? It's actually really elegant. Think about it this way. All you need, right, is a few different examples of the transporters on the left to keep pumping out protons to make this big proton gradient, right, of protons that want to flow back into the cell. Then you can have a huge assortment of different proteins that take advantage of that proton flow, but each of those move different types of solutes, right? So that's why indirect active transport is a very important way that our cells evolved, right? So that these transporters can take advantage of the energy that was used by the direct transporters to set up things like these proton gradients. All right. So many direct active transport pumps are what we call ATPases. Right? In other words, they bind ATP and they break ATP. Right? They break an inorganic phosphate off. Okay. So because the protein is designed to break ATP as its main sort of, function or its main mechanism, we say its primary role is an ATPase. Right? So in other words, it kind of generically just gets this name ATPase, which is kind of disappointing because you might say, well, this would be much more exciting if we called it a, proton transferase or something like that, right? But you realize the transferring of protons in this case is actually a side effect of the shape of the transporter changing its shape, right? And why did that transporter change its shape? Because its primary job is to break the ATP. It's kind of a weird way of thinking about it, but it's a great way of thinking about it because that means these proteins evolved to break ATP, right? And as a result, over eons of evolution, some other kind of side effect also evolved so that they can actually do things like pump protons, right? As a function that the cell then needs. If you look at this illustration over here, think about it this way, right? Because I know it's really confusing to think that well, why is this protein just called an ATPase? That's so generic. Imagine, for example, here we have this guy, he's trying to lift a car up, maybe to change a tire, or I guess maybe he's gonna, I don't know what he's gonna do here. But so he has this jack, right? So I think you all know the shape of the jack is suited for the fact that he can put in energy on one side to change its shape So that would be like a protein binding ATP to change its shape, right? And then another part of the protein changes its shape, and that's where the work is done, right? So why then are we satisfied just giving this a generic name like ATPase? Right? Because think about this. If there was no car in this photo at all, and it was just the guy working the jack, right? Would you say the jack is broken? No, the jack would actually still do exactly what it's meant to do, which is only to do what? To change its shape in according to how it's designed. Right? The fact that you can lift a truck if the truck is placed over the jack is a side effect, right, of the jack doing what the jack is designed to do. Right? That also means that if you were trying to lift something else, for example, instead of lifting up a car, maybe you have a whole series of jacks that are meant to do something like prop up the foundation of a house, right? Those jacks, right, might be shaped differently in order to properly do that particular type of job. But in principle, right, they would still be considered an ATPase. Energy is put in through ATP and they change their shape. Right? So this is an example of what we call a P-type ATPase. In other words, the phosphate group ends up being stuck onto the protein, and that's what forces it to change shape. But there are other examples of ATPases where the phosphate group doesn't actually end up being stuck on there. Why? Because as we looked at, if the function of this pump depends upon a phosphate group being attached to it, then part of the cycle of how this pump works also involves you having to take the phosphate off, right? And for something like a pump that needs to move very efficiently and very quickly, right, that might not be a good way to do it. Right? So this table summarizes different examples of these ATPases. So all of these are proteins that evolve to break ATP, that's what we call them ATPase, and they change their shape in a way that secondarily some other function is enabled, right, that the cell needs them to do. So on the top here, again, are the P-type ATPases. So P stands for phosphorylation. So the protein itself gets phosphorylated. So the pumps that we looked at, like that move certain ions like potassium, are examples of these. These would be found in the plasma membrane, right, the cell membrane. Other examples are V-type ATPases, V standing for vacuoles. So in this case, these ATPases, when they break the ATP, they're not stuck with a phosphate group on them. They just break the phosphate group, right, in order to get the energy released to be able to change their bonds, right? And so many of these are involved in actually causing the vacuoles or the vesicles inside of your cells to have things concentrated into them. For example, like protons. All right, so V-type ATPases, many of them move things like protons to actually change the pH of something inside of a vesicle. Another example of a proton pump are F-type. Right? We will learn about F-type ATPases later in the semester because the pumping of protons by these F-type ATPases is what enables other indirect transporters to tap into this artificial proton gradient to drive things like the production of more ATP molecules, right? Which is how our cell derives energy, right? And then the fourth group are what we call ABC type ATPases, ATP binding cassette, right? That's where the ABC comes from. These are pumps that are involved in using energy to pump certain things out of your cell or in, like nutrients they can pump in. But importantly, for pharmaceutical sciences, these are the types of pumps that pump things like drugs out of your cells, right? Because certain types of toxic drugs, things like that, if they get into the cell, maybe just by diffusion, because many drugs are small hydrophobic molecules, they need to be pumped out of the cell, right? Otherwise, the cell might actually die from having these compounds in them, right? So ABC ATPases are examples of those. Okay, let's look at an example of an indirect active transporter, right? So I said that indirect active transport is when you have a transport protein like this one here that's taking advantage of some maybe gradient that was set up, like a very strong proton gradient, high protons on one side, low protons on the other. This protein transporter is designed to be the right shape to allow these protons to flow back into the cell, for example. And in doing so, it taps into that energy and it does work in the form of moving this other solute molecule across against that solute's concentration gradient, right? So you can think about it as this water wheel, this wheelhouse here. You know, the water that flows over the water mill here, right, turns it, and inside the wheelhouse, there's some kind of work going on, right? If the water didn't flow over the wheel, let's say the water on the right here is too low, then you can't do any work inside the wheelhouse. Okay? So you might say then, well, I don't understand, because on the left here, where's the energy that actually goes into this, right? This would be an example of indirect active transport because it is tapping into a gradient that was set up here. Gradient meaning, in this particular case, the water somewhere else that you don't see in this cartoon had to be raised up in the first place, right? Into this sort of series of flowing, channels or whatever, so that they could then be allowed to go back down again due to gravity to drive work. So the fact that somewhere else, not shown in this cartoon, is a pump that's actively pumping the water up and raising it up, right? That's where the energy was directly used, right? And then you have something like this water mill here, right, that can indirectly take advantage of that energy, right, to actually have the work happen, okay? So here's an example in a cell of one of these indirect active transporters. We already looked at the transport of glucose across the cell membrane in the context of facilitated diffusion, right? We said most of the time you would have higher sugar, let's say flowing around outside of a cell, especially if you've eaten a big meal and you have sugar flowing through your blood, right? And the cells, they want that sugar. So the glucose can diffuse across the membrane, but because the glucose is too big to go through the membrane itself, you need something like the GLUT1 transporter that we looked at last time, right? That just binds the glucose on one side, changes its shape so the glucose can get into the cell. In that case, right, that was still being driven by high glucose on the outside wanting to flow downhill, down the gradient to the lower glucose inside of the cell, and that's how your cell would get glucose. But there are some cells in your body that actually have to actively move glucose in. Why? Because maybe in certain types of cells in your body, for example, your liver cells, right, their job is to concentrate glucose, to take all that extra glucose from your blood, right, and concentrate it and chain it together to make things like glycogen. Right? So in that case, those cells would actually require energy to keep moving more glucose into the cell if the concentration of glucose is already high inside. So that's where this particular example comes in. This is the sodium glucose symporter, and it's an example of indirect active transport. In this case, this sodium glucose symporter starts with being open towards the outside, okay? And it's going to move sodium from the outside to the inside, all right? Now remember, the sodium was being actively pumped out of the cell by the sodium-potassium antiporter that we looked at earlier in the lecture. So that was an example of artificially making, right, a disequilibrium, a high sodium concentration on the outside that wants to flow back in. So this particular purple symporter can take advantage of that. The sodium wants to flow back in, so it binds there. And at the same time, right, glucose on the outside can bind to the transporter at the same time that it's bound to the sodium, right? And the sodium wants to rush into the cell, so it changes the shape. And in doing so, it releases the glucose into the cell. And the glucose is being pushed against its concentration gradient if, for example, this is a liver cell that's busy concentrating the glucose. Again, the principle is the flow of sodium wanting to rush in because of the existing artificial disequilibrium of high sodium on the outside versus low on the inside. That force is stronger than any resistance that the glucose has in being shoved up against its concentration gradient, right? Okay. All right, so I just wanted to end with this last slide here. So I mentioned V-type ATPases as being ATPases that pump protons, right, into vesicles in your cells to lower the pH, right? These V-type ATPases are incredibly efficient and effective. They can create a 10,000-fold strong proton gradient across membranes in vacuoles and in organelles, right? How do they work? Well, think about this gumball machine here, right? In this case, ATP would be bound by this gumball machine, and this gumball machine would be the V-type ATPase, and the breaking of that ATP is like turning the crank on the gumball machine to allow things like gumballs to come through, right? And in this case, right, the shape of the gumball machine is not somehow temporarily modified by having something stuck to it, right? It's just the energy itself of breaking ATP keeps this crank moving, right, very efficiently to keep pumping protons through. So why do I use this example of a gumball machine, right? Because, here it is. This is the V-type ATPase illustrated on the membrane of a cell, right? So it's made up of different proteins. Each of these different lettered A, B, C, D, E, F, right, is a different protein subunit, okay? But together they form this remarkable machine. And as the top part here is binding and breaking ATP into ADP, right? That release of energy changes the shape and causes this bottom part to spin, just like the crank on the gumball machine. And that's what then passes protons through from one side of the membrane to the other, right? And you say like, well, this is just a cartoon then. What does this thing really look like? Well, I'm not going to disappoint you by not showing this to you because, I love this stuff. Here it is, right? This is the actual protein complex of this V-type ATPase, right? And again, each one of these colored subunits is a different protein, A, B, C, D, right, all the way through G. Think about this. Each one of these proteins is coded for by a different gene in your genome, right? That gets transcribed, the RNA gets edited, then the mRNA gets translated to make a chain of amino acids. That chain of amino acids, because it's surrounded by water, will fold up into just the right shape because of the identity of all the amino acids and their side chains that were strung together, right? And then these protein subunits come together in this truly remarkable structure, right? There's no denying. If I were to describe this as a molecular machine, I don't think anybody would dispute that, right? And the very fact that the shapes of these are just right to be able to bind and break ATP, causing the changes in the shape of the proteins to spin, right, thereby pumping protons through, I think is just a beautiful, beautiful thing, right? Okay, let's go ahead and stop there today, and I'll go to the chat. Okay, in terms of osmoregulators, like certain fish, are their cells constantly changing shape based on their environment, or is their cell a particular shape that can just tolerate all environments? Their cells pretty much have to stay the same shape, right? They can change their different osmolaric potentials based upon things like an array of different transport proteins that are busy doing things like regulating solutes, regulating water content, right? Because even though cells do change their shape as part of many normal cells functioning a certain way, right, there's only so much change in shape that a cell can tolerate before it loses its proper function, right? So it's more about doing things like being able to, if you have a dramatic change, let's say from salt water to fresh water, like a fish, right, that its cells can adjust by adjusting things like solute concentrations in order to compensate for that and keep the cells, the integrity of the cells intact. Here's a question. Do we have to know all these different ATPases? Well, you have to know that there are the 4 types of ATPases. So if that's what you mean, right? Yes, you need to know that there's 4 types because, we'll be discussing examples of them, right? So as a life science student, right, who appreciates the need to understand how cells work, a big part of understanding how cells can function in tissues is by understanding how they have these different types of ATPases to maintain proper things like solute concentrations. Do we need to know specifically how many sodium ions go into the sodium gradient? Yeah, I mean, so for the example of the sodium potassium antiporter, I don't think it's that hard for you to know that there's 3 sodiums that move one way and 2 sodiums that move the other, right? Okay. All right. So, one thing I wanted to mention also, now that I didn't want to take up time during the lecture to talk about this, but I think this is a really interesting thought for those of you who are still kind of on here, right? Cells, living cells as the basis of life on Earth, first evolved where? They evolved in the ocean, right? In a saltwater environment. So not only was the first experiments with organic molecules coming together happening in the context of water surrounding it, but it was salt water, right? Meaning that had to have been, taken into account. So all the choices of how cells evolve to function and survive, right, and deal with things like osmolarity and things like that, right, evolved in the context of not just water, but salt water. Right? So that's why life began in the ocean, and the single-celled microbes in the ocean could deal with that very easily, right? Because they just have their own membrane that they're moving stuff in and out of. But what happened once life moved from the ocean onto land? Right? After all, if 4 billion years ago, the first life on Earth, right, figured things out, meaning things like DNA, carbohydrates, polysaccharides, lipids, and how all these things were going to work together, but they did it surrounded by salt water, how could life move to land? Well, you could say, well, on land, there's pockets of water, of course, right? Ponds, lakes, and things like that. So again, simple microbes, you can imagine them adjusting to things like fresh water, right? But overall, how did life move to land in terms of big organisms like us? And I think the wonderful thought here, right, is that our cells still echo how these different mechanisms first came about when life was first invented 4 billion years ago, and these mechanisms haven't changed all that much. But we are not surrounded by salt water, right? Sitting here at my desk, you sitting wherever you are looking at this on the screen, right? So the key is we carry the salt water with us, right? So we are packages of the ocean, represented now by the salty blood fluid that makes up our plasma. When I showed you all those illustrations of the sodium and the chlorine ion components of our fluid, right? Maybe it didn't occur to you that in a way, that is like an echo of our ancient history, the fact that our cells evolved to need to be in a saline solution, right? So we are like the ocean packaged up on 2 legs that we walk around and carry a part of the ocean with us, right? And that's why water is so important. And that's why the salinity of your body's tissues are also so important. Okay, something interesting to think about. All right, so if there's no more questions, then we can go ahead and stop there today. So have a good rest of your Wednesday. I will see you on Friday. Okay, all right, bye-bye.